Hello, hello, testing. Okay. Testing my mic. Okay, so I think my mic is okay. Okay, so hello. Okay, so I think we can start our class now. So already 8 o'clock, so we start our class now. So okay. So yesterday we already learned about the process, the protocol of the genetic engineering, right? So for today, we will learn about the application that have in genetic engineering and also the cDNA and genomic library. So for today we will focus more about the application part lah. So all of the overall process of genetic engineering, I think you guys should already know it. What is it about? Okay, okay. So let us continue. Okay. Okay, so this one is just the recap about the genetic engineering process. So basically, genetic engineering is we need to modify the gene, right? So we modify the gene, and then this gene can produce the desired trait that we want. So, like for example, like make a person handsome. So you take a gene that can make a person handsome, so that when you insert into the plasmid and then replicate by the bacterial cell, then this cell will produce the trait, the protein that can make the man become more handsome. So like that lah. So normally the overall genetic engineering process, the first thing is you need to isolate the gene of interest lah, gene that we need, that we want, that can express the protein that we want. For example, like insulin. You will isolate the insulin gene and also the plasmid. So later you use the restriction enzyme to cut the plasmid, the restriction site of the plasmid and also the gene of the interest, to cut the fragment so that this DNA fragment will be inserted into this plasmid, DNA plasmid. Then it will form the recombinant plasmid. And then after that, it will undergo transformation process to the host cell. So normally the host cell is using E. coli. And then this E. coli, then the host cell will undergo screening process, because you need to ensure that all of the cell in the culture really containing recombinant plasmid. After we know that the colonies, we select the colonies that have the recombinant plasmid. After that, we will culture the solution. So there are two methods: either you using the modern one, PCR technique, or you using the traditional one, which is using the culture medium. You using the culture medium to culture the bacteria, so that this bacteria will be multiply more and more, produce more and more bacteria. So this process is what we call as cloning. And then this bacteria, because due to the presence of this gene of interest, so it can express the protein that we want. So normally it's enzyme or hormone lah, like insulin. So like if the host cell is containing the insulin gene, meaning that it will express higher amount of insulin lah. So this is the overall process of genetic engineering. So hopefully yesterday you guys got attend, you already master it. What are the process? So for today, we will not focus very more for the genetic engineering process. We will focus on application. So normally before we proceed to the application, so normally if you want to isolate the gene of interest, especially in eukaryotic gene, like animal gene, human gene, or I want to isolate fungi gene. All these eukaryotic gene, as we compare to prokaryotic gene, eukaryotic gene is more complicated and more longer compared to prokaryotic gene. So meaning that the eukaryotic gene we cannot directly use as the gene of interest. We cannot use directly. Unlike the prokaryotic gene, yeah, prokaryotic gene, if your gene of interest is prokaryotic gene, yeah, you can directly isolate it and directly insert it. Just directly continue to this process. But for eukaryotic gene, especially in human and animal, or not only human animal lah, human animal plant, all these is eukaryotic gene, right? They all contain eukaryotic DNA. So this one you cannot directly to isolate it. Why? Why cannot? So normally why? Because of genomic DNA contain intron. If you still remember that you learned in gene expression in last chapter 17, gene expression, about eukaryotic DNA, it contains two regions, right? Either it is intron or exon, right? And then actually only exon containing the DNA code that can be expressed to produce the protein. Intron does not contain any protein code. So meaning that in genetic engineering application, we normally why we want to that gene, meaning that we want that gene to produce protein, right? To produce and express the protein or enzyme or hormone, all of that lah. But in eukaryotic gene, they contain intron. Intron kami tak nak. Tapi when you cut the gene, you cut the gene, normally maybe you can cut about the intron part. So we need to increase the efficiency. We don't want intron. We need to ensure that all of the genes are exon. But how to ensure it? Because eukaryotic gene originally, yeah, it already containing intron. If you want to cut it, not easily. Not easy. Not easy. Because you need to know that all of this is human-made process. It is not a naturally process. You isolate the DNA, isolate the genome, it's all of the human process. Because in naturally, the cells cannot isolate the DNA naturally. Tak boleh punya. All of the process, you want to go to transcription process or translation process, all of this occur naturally. But for now, we need to human make. So how to ensure that all of the genes that we need is only contain exon. We don't want intron at all. So one of the method is we synthesize cDNA. We use the cDNA as the gene of interest rather than using genomic DNA. Genomic DNA is the original DNA lah. From you directly isolate the eukaryotic gene, the eukaryotic DNA, then that is called as genomic DNA. But if you do a modification process, that is we synthesize cDNA. So what is cDNA? So normally cDNA, it provides a direct representative coding information, meaning that the cDNA does not contain the intro region. It only contains the exon region, meaning you can directly use this cDNA as template, undergo conscription directly to produce mRNA, and then directly use this mRNA to translate the protein. So normally you see here cDNA specifically represent the mRNA of gene, meaning cDNA is actually a template of the mRNA already. Represent the mRNA of the gene already. And then, so it can be actively transcribed and translated into the protein through the gene expression process. So it will reflect the actual production of functional gene. So compared to you use the genomic DNA, so if you use the genomic DNA, there are the probability that when you insert this gene, you cut the gene or insert the gene, it maybe is intro one, not rather than exon yang kami tak mau, yang kami nak. Tapi intron kami tak nak. We need to reduce all the possibilities. So how to reduce it? We will use cDNA. So how cDNA is synthesized? This is a process. This is the process. This for all of this process is done in lab, are done in laboratory. This process will not naturally occur in the cells. Memang tak akan occur. The cells only will occur undergo transcription. We know that DNA can undergo transcription, right? We know that the original cell DNA will undergo transcription process, and then the eukaryotic gene itself also can undergo splicing process. When we undergoes all these, undergoes like transcription, undergoes splicing process, then that mRNA we need this mRNA because this mRNA is the RNA, the strand that only contain the exon region. All of the intron region are already spliced out already. So this mRNA is that we need. So meaning that we need to isolate this mRNA. So we not isolate DNA, and also we not isolate the pre-mRNA that directly transcribed from the DNA one. We only isolate the RNA that is after splicing one. After splicing, you confirm that after splicing semua tu hanya mRNA yang hanya exon sahaja later will be undergo translation. But for now, we don't want the cell undergo translation. We will directly isolate this mRNA. So how to isolate it? Normally, we will add one key enzyme is reverse transcriptase. Normally, reverse transcriptase added to test tube containing mRNA is isolated from a sample. So reverse transcriptase is very important enzyme. You isolate the mRNA using reverse transcriptase, and also this enzyme also very important to synthesize cDNA. Because we need to use cDNA. We're not using this mRNA as the strain. Why? Why we want to use cDNA? Why tak boleh directly use mRNA? Because you need to know that our plasmid is DNA. Our plasmid is DNA, and then you can DNA and RNA are totally different things. You cannot use it. You cannot say that mRNA can directly insert into the plasmid. Cannot. If you say this is totally wrong because plasmid is DNA, not RNA. So that is why that we need to synthesize it become DNA, not RNA. So we cannot directly use RNA insert into the plasmid one. So you need to modify again. You need to synthesize DNA. So that is why as we call as cDNA. So cDNA actually full name is complementary DNA. Why it say complementary? Because it is totally complementary from mRNA one. That memang complementary one. Because we use the mRNA as the template. So dia macam DNA replication process. It look like DNA replication process, but the difference is DNA replication you use DNA as template, right, to produce the DNA double-stranded strain. But for now, the template changed to the mRNA. Dia tukar template dia. Dia tukar template to become mRNA, and then it use this template to produce double-stranded cDNA. Just template tak sama aja. Compare to DNA replication dia guna DNA as template. But for now, cDNA punya dia guna mRNA as template, and the enzyme also totally different. So the enzyme it used is reverse transcriptase. So this reverse transcriptase, it will synthesize the first DNA strand using mRNA as the template. And also they are also using the mRNA as a template, and a short poly-DT as a DNA primer. So as we know that DNA is cannot directly added. So normally they are the primer lah. You will create a primer. This primer also created by ourselves. So the primer normally is poly-DT lah. Why poly-DT? Because you know that after splicing process, mRNA will contain polyatyl and also another code region, right? But another one is not important. Polyatyl is so important, so that you can create a primer which is poly T. So normally it's T or are T at the 5' end, and then after that you can use reverse transcriptase bind to the primer. After that, it will synthesize DNA. The DNA nucleotide will be added using the mRNA as template to synthesize. This one just synthesize single-stranded punya. Dia copy information tu hanya copy satu line je. Or one single-stranded DNA only. Okay, after that, once the single-stranded DNA is done, it already done read all of the information or from the mRNA. After that, mRNA will be degraded lah. Will be degraded because we don't want the mRNA already, because we already have the information. Then we already don't want the mRNA. Then we will use another enzyme, DNA polymerase. Now we will use the DNA. This single-stranded DNA will be act as the template. After that, it will synthesize another strand of DNA. So kerana DNA ada double-stranded, right? So single-stranded bukan DNA. So it will synthesize another strand of DNA using this DNA polymerase. So using this polymerase, so it will synthesize another stranded of DNA. After that, finally, you will produce the double-stranded DNA. So this DNA is known as complementary DNA, cDNA. So this DNA totally have the complementary information from this mRNA one. Apa yang mRNA ini ada sama saja dengan ini cDNA punya. Memang sama aja. Karena awak guna template origin your template is from mRNA. So meaning that this cDNA will be act as the gene of interest yang kami nak lah. Kami nak punya. Mana dia memang akan will be expressed. So after it will undergo expression, right? After undergo expression, it will form this kind of mRNA. Memang totally exactly sama. Kenapa kami nak synthesize cDNA? Because in plasmid injection, plasmid is DNA. So we cannot directly use RNA to insert it. Memang tak boleh. So that is why that we need to produce one type of one human-made type DNA. Baru boleh complete ini genetic engineering punya proses. Kalau tak, memang tak boleh. Kalau tak, memang tak boleh. Kerana RNA memang tak sama dengan DNA. Tak boleh gabung punya. Hanya DNA boleh gabung dengan DNA atau RNA gabung dengan RNA. Yang ini dua benda tak sama, mana boleh gabung, kan? Okay. So this is all about the cDNA synthesis. So this is the very important step during especially in eukaryotic gene. So when you mention eukaryotic gene, normally is human, human, plant, human and plant. Human, plant and animal. Okay. So normally the most popular one is using insulin. You will use insulin to produce human insulin production using genetic engineering, and this is the very famous exam question. You can refer to past year. Normally every 2 year dia akan keluar. Memang dia akan keluar. Memang akan keluar. So normally this is all the steps. So you will see here, examination memang awak kena tulis semua ni. Memang. So normally the step is same as that we mentioned. Just that original step you need to involve cDNA synthesis. You need to involve this process. After synthesize cDNA, baru awak boleh cakap semua ni. You cannot directly say that. If you directly say that, you isolate the insulin gene from human cell. Totally salah. Memang totally salah. You are totally wrong concept because you already skipped the process cDNA. Because I already mentioned human insulin is eukaryotic gene, so you need to synthesize cDNA first. So if you directly say that the human insulin gene is directly isolated from human gene or from the pancreas, totally salah. Memang salah terlampau salah. Kalau salah tu memang maka awak akan sangat rendah. Although you already write all this correct, but because of your first step is already wrong, so normally your marks is very low. Normally 10 marks you will get only 3 marks like that lah. 3 marks lah. Akan ada punishment lah, memang ada punishment punya. Kerana your first step is already wrong, meaning that you already wrong concept. Awak tak tahu apa itu cDNA. So memang tak boleh bagi awak markah tinggi lah. So I just go through this one by one. Okay, normally the first part is you need to mention mRNA of the human insulin. So this mRNA is specifically for human insulin. Is identified. We need to identify it. After that, we isolated from what? Insulin is created from where? Beta cell of islet of Langerhans of the pancreas. So you need to mention all this. You cannot just mention pancreas cell. Pancreas cell apa? Pancreas cell ada banyak. You know that pancreas got alpha cell and beta cell. Beta cell. So insulin is special for beta cell. This one is you should know already because you already learned in semester 2. You already learned at semester 2. So this one you should know. You should write this full sentence. If you like the pancreas only no marks because not specific. Okay. And then and then after you isolate the mRNA, I already say mRNA will act as the template to synthesize cDNA. So that the short nucleotide that act as the primer known as poly-DT, the polynucleotide punya T will attach to the mRNA. And then the enzyme known as viral transcriptase is added to mRNA and then primer will start to synthesize single-stranded DNA molecule. Okay, and then the RNA-DNA hybrid molecule is produced. Okay, so yang ni dia cakap macam ni. This one is what we call as RNA-DNA hybrid molecule lah. Okay, after it form, mRNA will be denatured by other enzymes. So the single-stranded cDNA is catalyzed by DNA polymerase to produce double-stranded DNA. So this cDNA that will act as the gene of interest to form the recombinant DNA. So ini first step until this eight step. Step number 1 and number 8 is just to mention the cDNA synthesis. That cDNA can act as the gene of interest. Belum sentuh about the process in genetic engineering. So kalau awak skip ni proses, memang maka awak hilang terus lah. Okay, after that you know that we need bacterial plasmid. So you need to isolate bacterial plasmid from E. coli. From the host cell normally is E. coli. And then after that you need the filtration enzyme. So this filtration enzyme will recognize the filtration site of cDNA and also the plasmid. And then it will cut cleavage the filtration site to produce the DNA fragment with sticky end. After that, when you have the fragment of cDNA, it will be inserted into the plasmid by using the DNA ligase to form the recombinant DNA. Okay, and then this recombinant DNA will be transformed into the E. coli. Okay, after that, the transformed material, I already said, this after transformation, you need to screening, right? You need to undergo screening process. So this screening process normally you will culture the E. coli in the medium. So normally it will contain antibiotic. This is the first screening lah. It will contain the antibiotic. So normally only the colony that you see the colony, meaning that bacterial cell contain the plasmid. If not, the bacteria will die. After that, you will culture the medium in agar, and then after that, you will produce the blue and white colony, right? And then white colony that we need because our gene contain LATZ gene. So if LATZ gene, within LATZ gene, we contain the filtration site, which the gene of interest will be inserted into the LATZ gene. So if the LATZ gene is absent, it will not hydrolyze the agar, so it will cannot form blue product. So the white colonies are the product that we want. So after that, the bacterial cell will be cultured in the fermenter or in the conical flask, all there. Okay, you will for cloning process. So this one, the bacteria will culture in this fermenter to to produce high amount of bacteria, and also this bacteria, since it contains the gene insulin, it will express large amount of the insulin in very short period. Lastly, this insulin will be purified because what is the cell culture, right? So you need to purify and harvest the E. coli, kami nak insulin tu je. So we purify this insulin. After we get this insulin, we can use it in medical field. So normally we were packaging it into the hospital or to the pharmacy lah, so that it can be used uh for the diabetes patient. So these are all about the insulin production process using genetic engineering. This is the very famous, and because of it is the first successful technique that using genetic engineering to produce this human insulin. So hopefully everyone understand it. Okay, inilah proses dia. Ini memang very famous one. Very famous. All of you should memorize it one. Kerana memang keluar punya. Kalau keluar memang untung punya. Kalau dia keluar memang untung. Because it very easy to score if you already know all the process lah. But if you still not know, I cannot do anything lah because I already teach it. So if you have any question, you just ask it. Okay, this is about human insulin. And another one is about using the human growth hormone. Another one is growth hormone. If you're using the human growth hormone, actually same process, more process, sama je. Just that first step, tukar lah. Bukan pancreas lah. Human growth hormone dekat mana? Human growth hormone, I think is thyroid kan? Thyroid gland. You isolate the cell from thyroid gland. Right. So, tukar kepada thyroid gland lah. And also, all the insulin turns, tukar lah kepada thyroid hormone yang kami nak lah. And then after that, all these are just, so normally all process are same. Just if using growth hormone, you just tukar the first step lah, yang paling penting first step lah. Don't say that you using thyroid hormone, you put say that it's isolate from pancreas. Thyroid hormone dekat pancreas ke? Bukan kan? So anyway, any hormone got not only thyroid hormone actually, but in your syllabus, the most important is insulin and thyroid hormone lah. Okay. And another in syllabus also in plant application in plant. Plant pun sama, but in plant we will know as gene transfer. Kerana plant ni because we produce plant, not the protein, this bacteria will not this bacteria only can express the protein, right? Express the enzyme. But in plant kami bukan nak express the enzyme, because in plant we need to directly dekat plant punya. Dekat plant punya. Kami nak transfer gene ni kepada plant, to the plant. We don't want just express the enzyme, all these enzyme only. Kami tak mahu ni enzyme. In plant, this enzyme, if it is alone, not meaning, because you need to transfer this gene to that plant. So this is what we call as gene transfer. You also can use this technology. So just as I mentioned yesterday punya tomato kan, merah punya tomato tanpa campur, when it contain the gene from the purple snapdragon, it can produce purple tomato, right? So the technique is using this. So in plant, it is slightly different when you do the gene transfer in animal or plant. But for your syllabus is about plant lah. It's slightly different that you learn at here. But the process still same. Just the final step may be a slightly different. Final step is not you enrich the culture in the media like that. The final step, the cloning step is different. But the previous step 1, 2, 3, 4, 5, step 1, 2, 3, 4, all are same. But the plasmid that we use is special, unique plasmid. We call as PI plasmid. Not using any plasmid. Bukan guna plasmid yang kami nak sesuka hati punya. We need to choose the plasmid that we call as PI plasmid. It is the special plasmid for plant only. So this plasmid normally can be found in one type of bacteria called Agrobacterium tumefaciens. Tumefaciens. Awak memang kena memorize ni, tak ada pilihan. You need to memorize. In human, insulin yang I always say is E. coli, right? As Herbacea coli. E. coli. Saya cakap E. coli, E. coli, E. coli. But in plant, not E. coli. In plant, the bacteria used is Agrobacterium tumefaciens. We use this bacteria as the host cell, and then the plasmid is using PI plasmid. Okay, so this is the structure of diaplasmid. This one you don't need to know lah actually for your syllabus. So this one I already mentioned. Agrobacterium tumefaciens as the host cell. So please remember it. Memang awak kena memorize semua. So yang ni, this one now become also very famous question. Famous question kerana human insulin dia keluar banyak kali lah. Dah bosan lah PM. So dia akan keluar soalan yang plant gene transfer. So normally the process is, you can see normally is same. Same. Normally, first step is you isolate the plasmid from this bacteria, Agrobacterium tumefaciens. After that, you use the restriction enzyme to cleavage the restriction site of the plasmid, right? And then at the same time, you also want to have the gene of interest. Same as the process that you used before, gene of interest. Awak kena gene of interest. So after you have the gene of interest, you will insert into this TI plasmid, to form the recombinant TI plasmid. Okay. After that, you will undergo the recombinant TI plasmid. Okay. Recombinant TI plasmid. So after that, you need to transform into the bacterial cell, right? Awak kena transform dekat bacterial cell. You will need to transform into the bacterial cell. I use this photo more easy. Yang ni lebih complicated sikit. Awak macam ni kan. Okay, you already form the recombinant TI plasmid, and then you transform this recombinant TI plasmid into this Agrobacterium tumefaciens cell, host cells. Okay. After that, you can undergo screening process, same as the screening process. Same as I mentioned in human insulin one, same process. After that, this is the final step, is totally different. This recombinant bacterial cell will not culture in the cell culture. Dia bukan culture dekat culture medium. It will directly transfer, the bacterium will be insert the will be transfer to the plant cell. You see here, the bacterial cell is used to insert tDNA carrying the foreign gene into the chromosome of a plant cell, meaning that this bacterial cell will be insert to the plant cell. Dia akan infect plant cell punya. This bacteria actually is the infection bacteria to the plant cell. Dia boleh buat infection kepada plant punya. So we use these properties to make a gene transfer into the plant. So that when this bacteria is infected, it will infect the bacterial cell by using the tDNA. Ini nanti saya akan mention. So this tDNA is that it already contain the gene of interest. Dia ada gene of interest dekat tDNA punya. So after you directly infect the plant cell, so this plant cell will grow in the cell culture first. Because this plant is very special. Dia bercabang menggunakan yang kami punya cara. Dia bukan bercabang Berapa guna yang old method. Dia it will not using the seed one. Dia bukan guna biji benih punya. We can directly use cell. So this one is about the plant tissue culture technology lah. So for those who study in biology in future, maybe you have the opportunity to learn about the tissue culture, whatever. But for now, you just want to know that this bacterial cell will infect the will infect the plant cell. After it infect the plant cell, so meaning that this plant cell already contain the plant cell DNA already contain the foreign gene dan gamina. Maksudnya, contohnya kalau macam ni, kami nak plant ni, I use the example from the yesterday lah, purple punya tomato. So the gene of interest is the purple color gene from the snapdragon. So this gene, when it occur in the plant cell, it can make the plant become purple color. So this snapdragon punya gene dia akan insert kepada tomato cell. So this tomato cell will already contain the gene from the snapdragon. Contain the purple gene from the snapdragon. So after that, you culture this tomato cell in the culture medium. After that, you will grow the plant cell will be grow. After that, you transfer back to the soil. After that, this plant will be grow normally, and then it will express a new trait. So for this example, it will express the purple color trait. So the tomato that produced from this plant is not red tomato, it will produce the purple color tomato. So I use the purple tomato case, so this plant cell is tomato. And then the gene of interest is the purple color gene that extracted from snapdragon plant. So this is the final step that is different from the human insulin. So this one is we call as gene transfer. So we transfer the gene into the plant cell. So we transfer the gene from the bacteria into the plant cell. So this is what we call as gene transfer. Okay, another one, this one I just want to tell you about that. The gene of interest, normally the gene of interest that inserted into the TI plasmid is we call as the region as tDNA. tDNA ni adalah region yang dekat TI plasmid that contain the nutrition site, so that the gene of interest will be at here. Gene of interest will be at here. So you see this guy bear tDNA dekat sini. It will add there. Okay, after you go to the it already at and this one is agrobacterium cell. So once this agrobacterium starting infect the plant cell, this tDNA will be expressed into the single stranded tDNA. And then this tDNA will be transferred into the plant cell. Ini teknik dia. It will transfer to the plant cell, and then this tDNA will be import. Dia akan directly affect the DNA structure of the plant cell. It will directly affect. DNA complex will import to the nucleus and make an integration. Meaning that yang original punya plant cell actually should be like the genetic code lah. I say that actually should be a genetic code for plant cell is AAA, TTT, CCC. But for now, when the tDNA is transferred, so this DNA already has gene of interest. So for example, our gene of interest is CCC ATCG. So CCC ATCG. So this plant cell yang original one dia sudah buang, lepas tu dia integrate into the yang gamina. So maybe CCC ATCG lah, CCC ATCG for already at here. So meaning that this plant already contain the DNA of the gene of interest. So meaning that it already have the gene of interest at here. So this is how the agrobacterium, once it infect the cell, it will transfer this tDNA kepada plant cell, and it will integrate the DNA sequence of the plant cell. So this is the process. So normally, if it does not contain the DI plasmid, does not contain gene control, actually it is one type of the disease that can occur in plant. Normally, it will one type of disease one. But for now, because we already take out the gene, so normally this one is what we call as gene transfer. Because the technique is macam ni. Ini memang macam occur naturally punya, bukan kami nak punya. Because in human kami buat back a vicious, we found that this eDNA can affect like this. So we use this kind of property to make genetic engineering so that we can modify the gene of the plant cell. Now, very directly, we directly affect the gene, modify the gene. So that when the this plant cell already have the recombinant gene that we want, then it will culture in the culture media. After that, when the plant grow, become the major plant, then this plant will have the characteristic lah, will have the trait that we want. So for example, purple color tomato. So ni plant memang ada purple color punya tomato. So okay, so that's all for plant. So plant is more spatial, especially in last step. Last step memang spatial sikit. So please remember it. Remember it. Okay. So that's all for genetic engineering part, application part. So that's all. You only learn three. Normally it's three application: insulin, growth hormone, and also implant. Implant. Okay, so animal so far not yet, but in animal actually the animal one is more complicated for now. So not you are not in the syllabus yet, but you if you study in you will learn it lah. Okay, the last topic is about genomic and cDNA library. So this one is about that we create one DNA library. So normally the genomic DNA library actually it is one type of library. Kami nak buat gene bank, genomic library, nak buat satu perpustakaan. You imagine perpustakaan, apa itu perpustakaan? Awak masuk perpustakaan, awak nampak apa? Banyak buku kan, wah banyak-banyak buku dekat itu rak kan. You can see many books in the rak, in the bookshelf, right? So this one same as here, meaning that we create a genomic sequence of DNA. You awak masuk je, you imagine that now you go to library, you are not see all the book, you see many DNA at there, many plasmid at there. Many DNA, different type of DNA, better than book. Awak pergi round, awak nampak ada DNA. Bukan buku lah, semua DNA punya. Yang ni lah maksud dia. So normally genomic DNA is that we using the directly isolate the gene punya. We directly. So normally this process is the first process lah. This is the first process. Yang ni. We just want to form the recombinant plasmid only. So meaning that the library is they have many different recombinant plasmid in one place. So when they are create many recombinant plasmid. So when scientists they are not so they create a many recombinant plasmid. So once a day, many 2 years ago, 2 years later, 3 years later, suddenly the scientists want to make a research again or want to find that I think I have made a recombinant plasmid A, but already 3 years ago. So you don't need to wasting your time already. You don't need to wasting your time. You nak cari, nak buat again, want to isolate again, want to make again. You don't need to wasting your time. You just directly go to the genomic library. You can directly find your recombinant plasmid already. Awak ada. So you can directly undergo transformation and then directly clone. Compare to awak nak buat lagi, nak restriction enzyme lagi, nak cut again, nak gene of interest, semua ni sangat susah. So you directly have the recombinant plasmid, you store at one place, so you can once upon a time, maybe 10 years ago, you want to use, yeah, you can directly use it because you already met it before. So this is what we L as genomic library. So genomic library. So the genomic library is set of DNA clone collectively containing the entire genome. So normally this one is we isolate the entire gene. Memang semua gene kami nak. We tak kisahlah dia exon ke intron ke, tak kisah. Kami semua nak. So we isolate all the gene. So normally this bank is more bigger lah compared to cDNA for one organism. So normally if you want to create a genomic DNA library for dog, for certain dog for dog. So you need to do many times of recombinant plasmid. Yeah lah, kerana you know that DNA library, the DNA is very long. So you need to cut all the gene is very difficult and very challenging. So it is more taking a time lah for compared to cDNA library. So normally you create the genomic DNA library. Normally same as the process you create a recombinant plasmid. So awak boleh tengok to create a genomic library, so normally you collect the cell. Memang like the job cell gene origin memang awak kena ada cell yang kamu nak. So and then you isolate all the DNA, all the DNA, and then this one we not choose the DNA, kami tak perlu choose, kami semua nak. Memang kami semua nak. Awak tak perlu choose punya. Then after that, the DNA is isolated, it is incubated with a filtration enzyme to cut the filtration site. So normally this one you don't need to choose the filtration enzyme. You don't need to you doing the genetic engineering for a certain purpose. Because this purpose is to want to create a library, so you don't need to very specify the filtration enzyme. You just choose the filtration enzyme yang plasmid punya ada, so you just directly cut and then cut lah. Whatever DNA nak cut, mana-mana cut sebenarnya. But for if you have a certain purpose, you need to choose carefully because not all of the DNA kami nak. But for now, for this purpose, we need all DNA. So awak tak perlu risau sangat nutrition enzyme yang awak nak guna lah. You can use any nutrition enzyme you want. So so you cut the nutrition side. So normally the cutting is very random because awak tak kisah awak nak apa-apa punya fragment awak tak kisah. So this meaning that the cutting is random. So the different DNA molecule will be cut at the different space. So maybe it can produce a overlapping fragment lah. So this is one of the advantages in DNA library. So maybe that in A punya you will see that A punya recombinant plasmid and A, B, C recombinant plasmid. Maybe A and B it has some gene is overlap. Like the A punya gene origin contain 1000 base pair, B will have 1000 base pair, but the within this 1000 base pair maybe got 2 to 300 is overlap with B punya recombinant plasmid. So this is one of the disadvantages of the genomic DNA library lah. But whatever, because genomic library we just create a library only, so we don't kami tak sangat pedulikan lah. Tapi when you want to do a research, it's more sakit kepala lah. Because they are overlapping, awak sakit kepala sikit lah. So normally the fragment then will join to the vector plasmid. Either you can use plasmid or bacteriophage, up to you. Okay, and then to form this recombinant plasmid lah. So this recombinant plasmid part of the gene, but most contain fragment that have no part of the gene of interest. So normally, that's all. When you form the recombinant plasmid, then that's all. Your genomic DNA library is already done. So normally, this one, the most unique characteristic is you directly isolate entire genome. Memang semua DNA awak nak. Bukan satu je. Bukan certain part je. Memang awak semua nak. Okay, compared to cDNA, so cDNA library, so the meaning is totally same right? Very simple right? cDNA, meaning you need to synthesize cDNA and then you store the recombinant plasmid as in cDNA form, not DNA punya. You not directly punya. So you store in the cDNA form punya. So apa cara dia? Cara sama je. Apa yang awak hasilkan cDNA sama je. Apa yang awak buat cDNA library. So the difference is cDNA library will only consist the sequence that are synthesized from mRNA only, meaning it only contain exon sequence only. Intron sequence you does not contain intron sequence. Awak tak ada intron sequence punya. Awak hanya exon sequence punya. So this one is more very useful in expressing compared to this. Because this one is very awak semua campur je. So when you want to do a research, want to maybe one time you want to do a research, want to find the enzyme atau gene sequence yang kami nak more difficult lah. But for this, very straightforward because it contain only exon, so you can easily to find the gene sequence that can express enzyme directly. So meaning that this more simple, more not more simple, more shorter lah. So the DNA sequence will be more shorter compared to this one, genomic DNA library. So the process want to form the cDNA library as same as the process that you form recombinant plasmid, just that you need to add on the cDNA synthesis process, just like that. Okay, and then this is the comparison between cDNA and genomic library. So normally I already say that cDNA library is a process to synthesize cDNA. So you will need to use the reverse transcriptase. Or the genomic DNA, because you just directly isolate it, so you don't need to using reverse transcriptase. You just directly isolate all the DNA that can found in the cell. But for this one, you only isolate mRNA. So cDNA only contain the sequence found in the mRNA. But for genomic DNA library, it contains entire sequence. So meaning that if you want to make a research that involve intron punya, memang awak kena cari information daripada genomic DNA library. But if your research is only involve exon, so you can find the information from this cDNA library lah. So one of the main difference is cDNA library only contain exon, genomic it contain both intron and exon. And also cDNA is very easy to express, so you can directly express already. So meaning that for cDNA, when you got that recombinant plasmid, actually you can directly to make a transformation already because it already contain exon. So meaning that you can directly undergo transformation and easy to be expressed. But compared to genomic, because it contain intron region, so maybe your recombinant plasmid 2 or recombinant bacteriophage is containing the intron. So meaning you need to do a splicing process. Memang awak kena buat splicing process. Baru awak boleh buat transformation. Kalau tak, memang awak buat transformation tak ada makna lah. Kerana awak punya recombinant plasmid is containing both region, maybe containing both region, or maybe even though your recombinant plasmid only contain intron. So you do the transformation no meaning lah because intron does not contain the protein coding gene. Dia tak akan hasilkan protein punya. And then another region, sequence is cDNA is more smaller and genomic is more larger lah. So there are the 5 differences lah. So these 5 main differences. So I think that's all for this chapter. So this chapter actually is very short chapter, but it is very important chapter. Normally it will come out in essay question. Essay question memang akan keluar punya. It normally in last question lah. Kalau dia keluar, memang dia akan keluar genetic engineering punya proses lah. Yang ni lah. Yang ni ataupun yang ni lah. Either lah. So hopefully you guys can understand it. So any questions? So actually the class is for today is until here lah. So any question? Think no question. Well, all of you very smart in genetic engineering. Okay, fine. So any question? So if no question, I will end the class today. So today class is about 1 hour only. So hopefully you guys, all of you know about the process in biotechnology, not biotechnology, in genetic engineering. So for tomorrow, I will go to the biotechnology, the last chapter of this semester. So okay, so that's all for today. So thank you and good night. Have a nice best. Bye.